this is the structure of insulin receptor. You can see that it has two alpha subunits and two beta subunits. So the alpha subunit and the beta subunit are interconnected by interchain disulfide bonds, and the beta subunit has a transmembrane domain, and it also has an intracellular domain. A very small part of the beta subunit and almost the complete part of the alpha subunit is present extracellular to the beta cells. Sorry, not to the beta cells, to all the cells, right? So the insulin receptor or the insulin binding portion is the alpha subunit, and the intracellular signaling is done by the beta subunit. So the beta subunit has a tyrosine kinase domain in the intracellular position. So these are the different receptors or domains that are present in the receptor, sorry, not the receptors, different domains that are present in the receptor structure. There is a large domain L1 and L2, cysteine rich domain CR, fibronectin 3 domains FN31, FN32 and FN33, and then you have the insert domain, that is insert in FN32. You have a insert domain for alpha chain and for beta chain, and then you have the transmembrane domain TM and the tyrosine kinase domain, that is the TK, right? So these are the different models that have been proposed by several research groups, and these models try to explain how the insulin receptor is activated. Alright, so the first A indicates what is called the yo-yo model, and in the yo-yo model the insulin binds to the receptor, and there is a sort of like string pulling that is happening, just like you're releasing a yo-yo, and a string pulling that is happening, which will activate the intracellular domain. The second model and the third model are called ligand induced models. In the second model, you can see that there is a cross. So the second model in the ligand induced second model, sorry, that is B, you can see that the receptor is in a cross linked manner in the transmembrane domain before the binding of the substrate, that is insulin. In the second figure, after the insulin binds, what happens is the cross-linking of the transmembrane domain is relaxed, and the cross-linking now happens in the intracellular domain. The third model, also called the ligand-induced model, explains that in presence of the insulin receptor, there is a cross-linking of the transmembrane domain, and as a result the tyrosine kinase domains come near to each other and it gets activated. The final or the D model is the rotation model, where it explains the rotation of the different domains, particularly the transmembrane domain, and it's almost a reversal of the B model. And only thing is that the binding of the ligand removes the flexibility of the extracellular domains and increases the flexibility of the intracellular domains. So these were the different models that explain insulin mechanism of action. So in this figure, you can see that you have the alpha domain and the beta domain, sorry, the alpha subunit and the beta subunit, and the insulin receptor transmembrane domains, as well as the tyrosine kinase domains. So the binding of insulin to the alpha subunit activates the dimerization of the alpha beta subunits, and the dimerization will activate the tyrosine kinase domain, which causes phosphorylation of the tyrosine kinases. And the tyrosine kinase phosphorylation will activate what is called the insulin response substrate, that is IRS, and the phosphorylated insulin response substrate will cause or will trigger the chemical responses or the cellular responses inside the cell. So in this figure, you can see that the insulin receptor phosphorylates two different types of proteins. One is the SHC protein, the other is the IRS proteins. So the SHC proteins and the IRS proteins are important for mitogenesis, protein synthesis, glycogen synthesis, and glucose transport. So as we have explained earlier, the PI3 kinase and the CAP-CBL combination will activate the GLUT4 or the glucose transporter exocytosis, not exocytosis like internalization into the membrane of the cell, plasma membrane of the cell, and it allows the influx of glucose into the cell. So these are the different biological functions of insulin. That is, it activates glucose uptake in muscle and adipose tissue, activates glycolysis, activates glycogen synthesis, activates protein synthesis, and activates uptake of ions, particularly potassium and phosphate. It inhibits proteolysis, ketogenesis, lipolysis, glycogenolysis, and gluconeogenesis. So all these processes are inhibited. So we have not seen the RAS-RAF pathway. So this is another figure which indicates the insulin resistance. So what is insulin resistance? So insulin resistance is seen in patients who suffer from the diabetes mellitus or adult onset diabetes mellitus, where insulin is being synthesized but the receptor is being increasingly non-responsive to the insulin that is present in the body. So that is called insulin resistance. So the insulin resistance is based on the two different arms of the insulin receptor. One is the metabolic arm, the other is the mitogenic arm. So the metabolic arm, which is activated by the IRS1, is important for anti-apoptosis, anti-inflammation, antioxidative stress, anti-lipolysis, glucose internalization, glycogen synthesis, and protein synthesis. Whereas the mitogenic arm is important in the RAS-RAF MAP kinase pathway, which is important for growth, proliferation, differentiation, atherosclerosis, inflammation, as well as hypertrophy. So, in cases of insulin resistance, particularly when there is hyperglycemia and hyperlipidemia, the insulin receptor metabolic arm is inhibited. So, when the metabolic arm is inhibited, there is an imbalance of the functions of the two arms of the insulin receptor. So, when the metabolic arm is inhibited, the mitogenic arm is activated more. So as a result, the anti-apoptosis, anti-inflammation, anti-oxidative stress decreases, and at the other side you can see that atherosclerosis, inflammation, and hypertrophy increases. So this will cause several complications as it goes further. And this is another figure which explains in detail regarding the PI3 kinase pathway. And the RAS pathway, you can see that you have the MAP kinase, the MAP kinase, and the MAP kinase. So these proteins are, these enzymes are activated in the IRS system. The GRB2 and the SOS are docking proteins which allows the RAS for like GRB2 is the docking protein for sauce, allows the sauce protein to bind to or interact with the IRS, and the source protein is important for activating the RAS-MAP kinase pathway. At the second time, you have the PI3 kinase, and the PI3 kinase pathway which activates AKT, and the AKT in effect affects several different proteins, inhibits several different proteins, and ultimately leads to cell growth, differentiation, cell survival, and protein synthesis. So this was about the insulin hormone.